Hi, thank you for having me. My name is Neeltje van Doremalen. I am a staff scientist at the National Institute of Health at the United States. And during the pandemic, we've been working on different vaccines against SARS-CoV-2. And today I'll be giving a short overview of what has been going on worldwide. So first, why do we need a vaccine? As you've been seeing throughout today, SARS-CoV-2 has infected over 8 million people worldwide at this point, and the global death toll reaches over 440,000 at this point. And in response, a lot of different countries have gone into lockdown. So obviously, lockdown is a temporary solution. It's not something that we can continue for the rest of our lives. And as a response, several countries have slowly started opening up already. The US is actually one of those countries. So by rigorous testing, contact tracing, and quarantine, we will be able to contain a pandemic to some extent, but we always risk the emergence of new outbreaks. And the outbreak that's currently occurring in Beijing is an excellent example of how we're not going to be able to contain it just via quarantine. So the way forward is really immunity in the human population. This is called herd immunity. We currently don't really understand what percentage of people needs to be immune to reach that, because we've never had to deal with coronaviruses in this extent. And more importantly, herd immunity is really not acceptable to be reached via natural routes. This likely will lead to excess deaths. We are seeing a large percentage of the people that are infected dying from coronavirus infection. So we really can't have that happening worldwide. So what we want to do is vaccinate people to induce this herd immunity as a safe response to the outbreak. So where are we with this currently? This is a slide I took from the New York Times that was recently updated. It basically summarizes how many vaccines are being developed and where they are. We currently have more than 125 vaccines in preclinical development. That means that they have not yet gone into human trials. They might be getting tested in animals, for example. We have 8 vaccines that are being tested in a phase 1 clinical trial, a further 8 vaccines that are being tested in a phase 2 clinical trial, which is basically just a larger clinical trial than a phase 1, still looking mostly at the safety of the vaccine. And then 2 vaccines are being tested in a phase 3 clinical trial. So that's a very large-scale clinical trial that's looking at the efficacy of the vaccine as well. But we currently have no vaccines that are being approved, and that's really where we want to go to. So before I go into any details of the vaccines that are currently published, I wanted to discuss the different types of vaccines that are being made. The first one is a virus vaccine. So that's either creating a weakened virus by passaging it through animals or cell culture. So this virus would still be able to infect people, but it wouldn't make them as sick. So you would get a potent immune response to the vector. That likely is less feasible in people that are immune compromised. We then also have inactivated virus. So here virus is inactivated, for example, by formalin treatment, and that could be injected as well. Usually you need an adjuvant to get a good response to that. The second approach is viral vectored vaccines. Again, you have 2 different types here. You either have a replicating viral vector or a non-replicating viral vector. The most famous example of a replicating viral vector is the Vsv Ebola Vaccine that is approved and being used worldwide now. Here, Vsv is modified in such a way that it's expressing the target of interest, which for coronavirus would mostly be the spike protein. A non-replicating virus vector would be adenovirus, for example. Here, certain proteins from the adenovirus, which normally causes a common cold in humans, are removed. So that means that the virus can infect cells, but it doesn't produce any infectious particles in the cell anymore. It will, however, the cell will still produce the target of interest, which again here is the spike protein, and your body will build up an immune response. The third approach is nucleic acid vaccines, and this is a very novel approach. It's either based on DNA or RNA, and the advantage of this is that they are very easy to develop and fairly cheap as well. So DNA vaccines need to be electroporated into the cells. That creates pores into the cells, and then DNA is deposited into the cells. It gets translated into mRNA, and that gets translated into virus proteins. Again, here you're looking at the virus spike protein that is the target. For RNA vaccines, there's a slightly different approach. The RNA is encased into lipid particles, and that merges with the cell membrane. The mRNA is deposited into the cells and directly translated into virus proteins. The final approach is a very classical approach. It's protein-based vaccines where you just express proteins of interest. So often, again, that's the spike protein. Sometimes that can be the M protein as well. You usually need an adjuvant to get a proper immune response to these proteins. A slightly different variant of that is virus-like particles, where again you express the different proteins, and together they form something that looks like a virus, but it's not actually infectious by any means. It's supposed to trigger a very strong immune response, but these can be a little bit tricky to make. So that brings me to the data that has been published so far. The first one I want to talk about is Sinovac data, where they used a whole inactivated virus. So they produced enough virus to be able to infect rhesus macaques. That got inactivated using beta-propiol actone and got mixed with an alum adjuvant. They then vaccinated rhesus macaques at day 0, 7, and 14, so 1 week apart, and they challenged the animals 8 days later. And that challenge was intratracheally, so what they do is directly deposit virus into the lungs of these animals. So you see two different graphs here. The first one is binding antibodies on the top, so that's measured via ELISA. And then on the bottom, you have virus neutralizing antibodies. These are thought to be particularly important to stop infection. So when we focus on the top panel, we see that binding antibodies are induced already after a single vaccination, but really we find a lot of antibodies after the 3 vaccinations in the rhesus macaques. You also see a slight difference in the high dose group, which is 6 micrograms per dose here, and a lower dose group, which is 1.5 micrograms per dose here. What is interesting to see is they also looked at antibodies after challenge. So this is 7 days after challenge. And you see a small increase in endpoint titer of binding antibodies, which suggests that this vaccine probably doesn't provide sterile protection. And I wouldn't expect that with the coronavirus vaccine. So that's not necessarily a problem. Then when we look at virus neutralizing antibodies, this graph was a little bit hard for me to understand at first, so I'm going to walk you through it. The green bars and black bars are the control animals, and then we've got our different days here. Then the blue groups are the low dose, and the red bars are the high dose. For blue, you see 2 different bars here. The darker blue includes this outlier here, which got a very high immune response. The lighter blue here excludes that outlier. Most importantly to take home from this graph is that this vaccine is able to induce virus neutralizing antibodies. And upon a 3rd vaccination, they are fairly high as well. Again, upon infection and 7 days after infection, you see that these virus neutralizing antibodies have increased, which suggests that we are not looking at a sterile protection by the vaccine. They then look at virus shedding via throat swabs and rectal swabs. Green and gray are your control animals again, so you see nice shedding in these animals. And interestingly, in our vaccinated animals, you don't see any viral RNA in rectal swabs at all. For the throat swabs, you still see some viral RNA in the vaccinated animals, but it is a lot less than what you're seeing in the control animals. What's very good to see here is that when you look at the lung tissue, whereas the control animals do have a fair amount of viral RNA in the lung. In the vaccinated animals, you find absolutely nothing in the high dose group, and there's a little bit left in the lower dose group, but it's significantly less than what you are seeing in the control animals. So that's very encouraging. This is the second vaccine I wanted to talk about. So this is based on DNA. They actually produced 6 different types of vaccine. They used one that is the full spike protein, and then they use different variants of that. So, for example, they have S1, which is just the first part of the spike protein, and it encodes the receptor binding domain, which is also called the RBD. They have a vaccine that is just the receptor binding domain, which is supposed to induce virus neutralizing antibodies specifically. And then here, the very last one is one that is basically stabilized at pre-fusion state. So spike throughout infection, it undergoes transformation of the spike. And it is thought that before fusion, the pre-fusion state is really needed to induce virus neutralizing antibodies. So these people did 2 vaccinations in rhesus macaques without an adjuvant, 3 weeks apart, and then they challenged the rhesus macaques 2 weeks later, day 35. And unlike the previous group, they used 2 different routes of challenge, the intratracheal route and the intranasal route. So it's directly into the lungs and directly into the nose where they deposit virus. Again, looking at binding antibodies here, we see that most of the vaccines are able to induce binding antibodies, although some of the animals do not seem to have a response to the vaccine. And then here on the right, we are seeing virus neutralizing antibodies. So you are seeing a response overall in these animals. And I'd like to point out that actually the pre-fusion protein doesn't seem to make a difference with just full-length spike, which is very interesting. So they looked at 2 different types of samples in the animals. They looked at a bronchoalveolar lavage, which is basically where you wash the lungs and look at viral RNA. So that's depicted over here. On the top, you see the control animal. So we see a lot of viral RNA in the bowel fluids. And then on the bottom here, you see the 6 different vaccines. So there definitely is a difference in viral RNA in bronchoalveolar lavage fluid in the vaccinated animals. There do seem to be differences between the vaccines. So the spike really seems to be the most promising vaccine here. What they actually measured is not just virus RNA, it's a subgenomic RNA, which is a measure of replicating virus. This is quite promising again. They then looked at nasal shedding as well. So on top, you see the control group again. You see very high nasal shedding here. And then on the bottom, you see the vaccinated animals. Again, they're looking at subgenomic RNA. And this is definitely reduced in the spike group. It seems to be reduced in the other groups as well, but maybe a little bit less for some than for others. So again, this is a very promising result. It really suggests that the vaccines are reducing virus replication. So then the third vaccine I want to talk about is a virus-vectored vaccine. This is one that I've personally been working on. It's a simian adenovirus vectored vaccine called ChAdOx1 nCoV19. What we did is a single vaccination in rhesus macaque via the intramuscular route, and then we challenged 4 weeks later at day 28. And we challenged using 4 different routes. We used the intratracheal route, the intranasal route, the oral route, and the ocular route. That's with a very high dose of virus as well. So when you look at binding antibodies, you see that those are being induced upon vaccination. And here you look at virus neutralizing antibodies, and those are being induced upon vaccination as well. We euthanized the animals 7 days after challenge, and here you're looking at lung tissue. So we were able to find lesions in the control animals, but not in our vaccinated animals. These lesions are filled with edema. We find syncytia cells in there and type 2 pneumocyte hyperplasia. And also, when we look at immunohistochemistry, we are only finding positive cells in our control animals, but not in our vaccinated animals. When we looked at bronchoalveolar lavage fluid, and we either look at just viral RNA or subgenomic RNA, we find that greatly reduced in vaccinated animals in rats. But when we look at nose swabs, like the previous group, we are still seeing quite a lot of viral RNA in the nose swabs. Lung tissue, when we look at viral RNA there, we see that it's greatly reduced in our vaccinated animals. So then the final vaccine I want to talk about is produced by CanSino. This actually is a phase 1 clinical trial, so that's very exciting that they published the first results here already. Again, this is an adeno-vectored vaccine based on adeno5, which is a human adenovirus. What they did is a single vaccination in 108 participants, and they tested 3 different doses, a low, middle, and high dose. And none of the participants reported any serious side effects. And this vaccine was able to induce binding and neutralizing antibodies. And what we're looking at here is a cellular response, specifically for T cells. So you can see that 2 weeks after vaccination, we are seeing these cells being induced, which is very promising. So in conclusion, we see that many vaccines are currently in development, and the initial data is promising. It might be that we are looking at partially protective vaccines. It needs a little bit more research to really know whether that's true, but it looks like some of the non-human primate models, we definitely see shedding from the upper respiratory tract still, but not necessarily any replication in the lower respiratory tract. So that could mean that you don't end up with pneumonia in a hospital. Instead, you just have a common cold, which is a massive win of what we are currently dealing with. For me personally, I think it would be very interesting to look at the immune response in older individuals, because that's really one of the risk groups that we are looking at. And timeline-wise, some of the groups, including the ChAdOx one, are promising vaccines to be approved as early as September. That is dependent on a lot of different factors. So we will have to see whether that pans out, but that would be much faster than we ever have done in the past. So with that, I will stop here, and I'd be happy to take any questions.
Thanks very much. Hi, Neeltje. How are you? Managing well with coronavirus. Thanks very much for your talk. Very interesting. And I must say, this is an exciting time for people who are interested in developing vaccines, because there's a huge challenge. And as you say, they've been now trying different routes at the same time, each one almost competing with the other, which is probably in some ways good. Maybe sometimes they should be working more together, but that's another issue. So I'll just ask a couple of questions. One is starting with the inactivated vaccine. We've had just the inactivated virus vaccine. We have good vaccines, the hepatitis A vaccine, the polio vaccine. So what are the issues about production of that vaccine? How easy is it to scale up production to the amounts that would be required to vaccinate virtually the whole world?
Right. So I think that's going to be the main bottleneck that you run into with an inactivated SARS-2 vaccine. It definitely doesn't grow to very high titers. So I imagine that would be a massive operation. Also, one of the things that you might run into is obtaining mutations in the virus itself that might reduce antigenicity of the vaccine. So I think that's going to be an interesting approach, but it definitely has its issues.
The other issue, which I think you just touched on, is the whole issue of basically putting these vaccines through an accelerated process. How concerned are you about the possibility that there'll be some unexpected, shall we say, side effects that are not picked up in the early stage?
Yeah, that's a really good question. And obviously with SARS, the first SARS that we saw, that was something that we did see in animal models. So with the clinical trials right now, they couldn't go forward until we had data in non-human primates in which we did not see any immune-enhanced disease. I think what we learned from SARS-1 is that usually is either inactivated virus seems to be more likely to cause an immune-enhanced disease. And there appears to be an association with the presence of nucleoprotein as well. So I think for vaccines that contain either of those, we should be on the lookout a little bit more for immune-enhanced disease. What we saw, so the study that we did in the non-human primates I talked about with ChAdOx1 specifically looked at immune-enhanced disease, and we saw absolutely nothing in those animals. Granted, that's 7 days after vaccination, and I'm sure we can make changes in our models to see if we can induce it somehow. And those are things that we are attempting to do. But I think clinical trials are going to tell us a bit more about that. So far in the clinical trials, we haven't seen any negative side effects of the vaccination yet. I'm not sure how many people that are vaccinated actually were exposed to virus as well. That's obviously a difficult question to answer at this point. It is a little bit concerning. Based on what I've seen in animal models, I'm not super concerned about it, but I don't want to exclude it either. And I think it's something we definitely should pay attention to.
The third one is that we're actually in this interesting situation where children seem to be very little affected by the virus. I mean, they get, not only do they seem to get a much milder disease, but there does seem to be evidence that they're not very active in spreading the disease, either amongst themselves or to others. I mean, this is at least the data that's been presented, which in a sense says that we're in this strange situation where the children would not be a target group for the vaccine. In fact, we would be targeting basically adults and probably the elderly. And the elderly are notorious for not responding well to vaccines. So you may have a vaccine, as you said, that's partially effective. And in elderly people, it may be very limited. Is that a possibility?
Yeah, I mean, that's a very good question. So the clinical trials right now that Oxford is running, they've got different age groups. So the very first group was 18 to 65 years old. And then you got, 55 years old, sorry. And once we didn't see any safety effects, we went from 56 to 69. And now they're actually testing over 70 years old. And one of the aims there is to look at the immune response. So I can't answer that question yet. I don't think anybody can, because we simply don't have the data yet. But it's definitely something to look at. I'm very much looking forward to that data. I think it's a very important question.
So the one point you made about a possibility that a vaccine or one would be registered or licensed in September or whatever, licensed for who? I mean, I'm saying something to you that there's a big difference between licensing and actually having an impact in the population. As we know, even the influenza vaccines are sometimes not as great as we hope them to be. So, I mean, but that's an old technology of inactivated vaccines that are sometimes live. But I guess isn't this going to create a lot of, I mean, you're probably not too much involved in the sense of the public response, but we're going to be actually, in a sense, saying there's a vaccine available, it's been licensed, but being licensed doesn't mean that it's that effective. It may be effective to some extent in some people some of the time. I mean, is that going to be a good message to give to the public? That's a hard question.
Yeah, it is, and especially with the anti-vax population that is out there, and it's particularly in the US actually very prevalent. Right. I think we need to be careful about how we approach that, and that's why so the Oxford group is the one that thinks that they might have a vaccine in September, and that's depending on a lot of different factors. So that's like the most positive outlook, really. But they will be doing a phase 3 clinical trial, so they will be able to say something about the efficacy of the vaccine. So it's a bit odd because they basically need transmission of the virus in the places where they are doing phase 3 clinical trials, which is the UK and Brazil and South Africa, and now they're moving to the US as well. So it feels a bit contradictory that we almost want transmission to occur.
Yeah.
We're hoping, well, they are hoping to include so many participants that we will be able to say something about the efficacy. And I don't think we should be giving vaccines to people of which we have not shown efficacy yet.
Right.
I think that could be potentially just giving the anti-vax movement more material.
The last thing is that, and I'm not sure that you're dealing with it, and that is passive immunization, using it for prophylaxis. I'm not talking about for treatment. I'm talking about using immune globulin to give partial protection, or at least protection for a limited time, which could be used as we have done for like hepatitis A, where it's actually been given virtually annually to some groups who happen to be, the military used to get it very frequently, to give them protection over a few months. Is there any interest in the, or there's interest, I know, but is there any potential in producing it in enough quantity? Again, this I guess is not something you're working on, but you may be involved a little in it. I don't even know.
So you're right, I don't really work on that. We did do some work, this is a few years ago, in marmosets using MERS-CoV, in which we treated them, but that was therapeutic, it wasn't prophylactic,
right,
with monoclonal antibodies, convalescent serum. And we infected animals with a very high dose of virus, and it didn't make much of a difference at all.
Right.
We did see some effect, but like that, those models are so different from the human situation, because again, we infect these animals with millions and millions of infectious virus particles. Hopefully that's not what you get exposed to in a real life situation. So it might help. What I understand from monoclonal antibody production is that it's still very expensive. So that might be more of a limiting factor.
Sure. Okay, I think that we have some more questions, but I think we'll leave them to the discussion afterwards. I really enjoyed your presentation. Thank you very much for the update. All the best.